Sarah Weckhuysen gets the award for coming a long way and getting to make that trip almost twice. It has something to do with Canadian airspace. I'll leave it at that. I actually don't know much more. Sarah Weckhuysen, direct from are you straight from Belgium?
Yes,
direct from Belgium. All right.
Thank you. Thank you, Sarah, for the introduction. I see, by the way, I still have the title slide from the talk I had to give yesterday. And thanks again for Ed. To Ed, I don't know where he is. Hi, Ed. Thank you for taking it over. I just shortly want to talk about a project that we're doing in our lab that I meant to include in my talk yesterday, but now that I'm here, I can bring it to you personally, because it's actually thanks to parents that we could set this up in our lab. So I owe it to you guys to tell a little bit about it and about what we're doing. Just as an introduction, you had a lot of talks yesterday talking also about novel drugs. But I think what is clear in general that there's still a need for novel treatments for this KCNQ2-related epilepsies. And then I think not only for treating seizures, but also trying to see if we can influence in some way the developmental problems that we see in all the children, and that are a real burden, of course, even later in life for all of the children and grown-ups that are also in the audience here. And as you probably heard yesterday and at talk, what has mostly been done until now is we've been testing mutations in a dish in cells, often oocytes, which of course is not the brain. And it tells us a little bit about excitability, but it tells us very little about what happens in the brain and in development. So what we need when we try to develop novel treatments is that we need models in which we can understand mechanisms of the disease better, and in which we can screen novel drugs and look for influence on development. And we can screen these drugs blindly. We can put a library of all different drugs on it, or we can really develop a hypothesis and try a novel drug on these models. Now, what can such a model be? It can be an animal which does show some behavior. And as you've heard, there's several mouse models being developed now, and there's more information to come in the coming years for sure, and we will be able to use these mice. But you can try it also in a little bit more basic model, and that's again in a dish. But instead of looking at an oocyte or an individual cell, you can really look at neuronal networks. Now, where do you get them? You can, of course, not just operate the brain of a child and take some cells and grow them in a dish, but there's luckily some other methods out there that have been developed in recent years and that have been used more and more by researchers. And this is something that we're doing in our lab now. What we're now able to do, I don't know, is there a... Yeah. So what you can do, you can take some cells from a patient. It often they start from skin biopsies, but nowadays you can also start from blood, and you can reprogram these cells into stem cells, pluripotent stem cells. And these stem cells you can drive to become any cell type you want, and one of them is a nerve cell. These stem cells you can freeze, you can put in the freezer for later use, or you can use them directly and start to grow nerve cells in a dish. This is, of course, still not a brain, but at least you look at neurons, at least you look at networks, and you can study them, you can put a drug on it and see what effect it has. Now, we're doing this in our lab and with one purpose, and that is to develop a novel treatment. And this is a little bit our hypothesis. Now, we have been talking in the last 2 days about KCNQ2, and as you have all heard, we all have 2 copies of KCNQ2, one from mom and one from dad, and one of them is carrying the mutation, as you see here. And we know from clinic, from looking at patients and testing mutations, that if you lack one copy of the gene, you get this benign phenotype, this benign familial neonatal seizures. So these are children that have seizures early in life, but then they develop totally normal. So it's, of course, frightening when you have a newborn with seizures, but at least later epilepsy goes away and children develop normal. In case of the severe mutations, and Ed has nicely discussed this just previously, these are different. So these are mostly missense mutations that either cause this dominant negative effect, so they interfere with this good copy of KCNQ2, or you have these missense that lead to a gain of function. So what is our theory? What if we could delete or silence this bad copy of KCNQ2? Then you would go from the situation of the severe mutation to the benign situation. You would delete one copy of the gene, and ideally or hypothetically, you would generate a situation in which there are neonatal seizures but no normal development. And this would work for both types of mutation, the dominant negative ones and the gain of function ones. Now the question is, of course, how do you delete or silence this bad copy of KCNQ2? Now, well, there's strategies that also have been developed by researchers in recent years, and this is something what they call, in general, RNA therapies, and one specific form is using what they call antisense oligonucleotides. And the antisense is referring to the fact that it's complementary to the RNA that's in our body. If you look at the first slide, or the first part of the figure here, so this is DNA, and you have a mutation at one part, in this case in KCNQ2, and this DNA, it's transcribed in a messenger RNA, and this in turn will be translated into the protein. Now, if there's a mutation, it will be translated in a mutated protein, and in general, all the drugs that we develop now, or most of the drugs, they're targeting this protein. For example, the potassium channel openers, retigabine, ezogabine, they were all working on the protein itself. Now, what you can try to do is to develop an antisense RNA that is complementary to this messenger RNA with a mutation, so that it traps it and that it gets deleted. So if you could do this, you could delete the bad copy of KCNQ2, because we're now able to design these antisense oligonucleotides in such a way that it only traps the copy with the mutation. So this is the aim. And this is not some fantasy or whatsoever, because if you... Oh, have another slide in between. Because, as I said before, you want to target this bad copy of KCNQ2. Now you can design ASOs for every specific mutation that's of all the children around here, but this would be a lot of work and a very costly thing to do. But there's an alternative strategy to this. Because all of us, we all carry variants in all our DNA, because if not, we would all be the same. And some of these variants, they are very frequent in the population. So it's a change somewhere in the DNA that does not have any effect. It just makes us a little bit different among us. And for example, in KCNQ2, there's one variant. They all get a name with a letter and some numbers that are very frequent in the population. This particular variant is present in 60% of people. So statistically speaking, probably also in 60% of children with KCNQ2. And now what we can do, we can test whether a child with KCNQ2 has this specific common variant, and we can check whether it's on the same copy as a mutation. And then we can design such a antisense oligonucleotide that binds this particular common variant so that the mutant copy gets silenced. So this is the hypothesis, this is the strategy. And again, it's not fantasy, it's not science fiction. This is a slide that comes from the website of one of the biggest companies that is developing such antisense oligonucleotides. And as you see, this is what they call their pipeline. These are different phases of clinical studies. And when it gets up to the end here, it means that it's really on the market. For example, this first drug, Spinraza, it's a drug that, at least in Belgium, a few weeks ago, it got reimbursed. And it's for a very severe neuromuscular disorder. But if you see below, these names of these diseases, Huntington, you might know ALS, Alzheimer, dementia, ALS. So these are all disorders of the central nervous system, of the brain. So this means these are drugs that are in development already for brain disorders. All of them, and that is still, I wouldn't call it a problem, but maybe a disadvantage, is to get these drugs into place. They have to deliver them intrathecally, so with the lumbar puncture that many people that had once an operation might recall. For example, all the ladies that had pregnancy and the delivery. So it's a disadvantage, but it's not unovercomable. And there's many research going on ongoing now to see if there's other way of delivering these drugs. So in a few years, we might be in a more advanced stage. So with the help of you all, this is what we're doing in our lab. This is the PhD student, Nina, that's working on it. What she's doing now, she's growing these neuronal cultures derived from patients' blood in a dish and studying them. We now are looking at cultures from healthy persons or healthy SIPs of patients. We have the blood and the neural culture from one child with the benign form and of several children with the more severe mutations. She's currently studying them and characterizing them, both electrophysiology, so looking at hyperexcitability, gain and loss of function, but also morphologically. They see whether these neurons, they make networks just as the normal in healthy persons or whether there's any difference. And what she's doing next is she will test these antisense oligonucleotides that we will develop to specifically target these mutations. And we will see whether, at least in a dish, we can turn the phenotype or the situation of the severe mutation into the one like we see it in children with a benign form. Of course, and I have to, without saying this is the miracle treatment, we have to put this in perspective. We're not there yet, even if this works in a dish, there's still some unanswered questions, because KCNQ2, it's already expressed early during development, and during embryonic development. It's still there when children grow up, and the question is, can we still influence KCNQ2 pathology later in life? So it's a very relevant question. This is one example in mice, in another disorder, that we might be able to at least influence it in some way. But again, it has to still be proven for KCNQ2. This is an example in another disease in which another gene, MECP2, is duplicated. It's the same gene that is mutated in patients with the Rett syndrome that you might have heard before. What they did, they made mice with a duplication, like in children. These mice, they're hypoactive, they have motor abnormalities, social behavior deficits, a little bit like in children with this disorder. And shortly what they did, they also developed these kinds of antisense oligonucleotides, and they infused it in the brain of adult mice. And they could see that they could revert these behavioral and intellectual deficits in these mice. Again, this is in another disorder. It still has to be proven for KCNQ2, but it at least suggests that there might be still some window for influence of neurodevelopmental problems. So again, the thing that we're doing in our lab, it's a proof of concept study. It's in a dish. It's on neurons, so it's already better than on a pure oocyte, but still. We will need further validation in an animal model, and we're working on that together with Ed. And specifically to answer these additional questions. How much time do we have to influence development? It seems logical that the earlier you start a therapy, the better, and that's why this initiative of Ed to diagnosing patients very early is very important. But the question will be, can we still win something later in life? And then other questions like, how long do we have to treat? Do we have to treat only early in life when the brain is developing? Or do we have to treat them lifelong? So all questions still to be answered, but I think an interesting path forward that we can do together with your help. Thank you.
I might be totally off, but I have to ask. I feel like I remembered hearing something a while back about radiation therapy that could target specific genes.
Regeneration or what's
Radiation.
Radiation therapy. I have to think. I know with radiation you can introduce mutations, but to correct them...
Or eliminate them.
The thing is that you will need to correct it, not just
Oh.
getting away, or at least for KCNQ2, you still want at least one good copy. So
Oh,
you don't
only
want
wanting to get rid of the bad one.
Yeah, if you get rid of everything, it's probably lethal. We don't know of any patients that don't have any KCNQ2. So,
Do you have any sense of how specific the ASOs have to be for the mutant allele versus the wild type, and how do you actually come to determine what that required selectivity would be?
yeah, that's a very interesting question. As I said, we want a big, we want some kind of selectivity, because as I said, we know that if you don't have any KCNQ2, it's probably lethal. We don't know of any children or human around that lacks both of them. So, and we also know that if you have less than 50% current, that you have some kind of disease and disorder, of severity. The question is, do we have to get all the bad KCNQ2 mutant KCNQ2 away? That's still a question that remains there. I think it might be that increasing or deleting, silencing it by 30%, it's enough, and this is something that we still have to test. What the amount is that we need to target. But some kind of specificity, it's certainly necessary, because we need the good one. The question will remain, do we have to take away all the bad or the mutated KCNQ2? Yeah.
I'm wondering about your iPSC study. Are you able to distinguish the BF&E neurons from the KCNQ2 epileptic encephalopathy neurons in the culture?
Yeah, that's the thing that we're currently doing. Not only electrophysiologically wise, and because this is something that we clearly already see in a single oocyte, that there is some difference, but we specifically also want to look whether there is a difference in, for example, network formation between all these neurons. So this is ongoing work, and I hope to be able to present it to you next time. We expect that there will be a difference, but this is exactly what we want to show, also to later show that when we use these ASOs, we can revert it from the severe phenotype, as we call it, to the benign phenotype. Yeah.
Very, very exciting. Thank you, Sarah. With the Spinraza for spinal muscular atrophy, my understanding is you have to give the intrathecal antisense oligonucleotide every 3 months. Yeah. Do you have any sense of how often you might have to give this, and how long it might last? I mean, you know, that will make a difference to palatability, I think. Too.
Yeah. Yeah, that's a good question. These are studies that would have to be done for every single disease, but we might expect that it's somewhere in the same range as, for example, Spinraza, as for KCNQ2. We see also, for example, in the mice study on MECP2, you see that after 4 to 6 weeks that the effect of ASOs at the level of protein expression is reversed. So the protein expression is again back to normal, which implies that you have to repeat it. There's other strategies that are a bit similar that use short hairpin RNAs and that deliver it through viral vectors, and then it gets inserted, and it would be a once alive thing, but it has bigger safety hurdles, because if there's any off-target effects, you cannot reduce them. So the advantage of this approach is that if it has any adverse effect, you can stop it, which you cannot with short hairpin RNA. There's other delivery methods that are being developed, and then it might be possible to extend the working time. But for now, it looks at it's 4 to 6 weeks, you see a real decrease in protein expression, or increase in protein expression. Yeah.
Thank you so much, Sarah. Lovely to see you. Thank you so much, Dr. Weckhuizen. And I know you went to a lot of trouble to get to us, and we very, very much appreciate it.